Okay, so one of the important things to pay attention to when it comes to non-disjunction is really the difference in the probabilities of having a child who's affected. And those probabilities change depending on if this non-disjunction occurred during meiosis 2, where the sister chromatids failed to separate, or during meiosis 1, where the homologous pairs fail to separate. The resulting gametes that occur really set up for a very different probability for whether or not your child will be affected, depending on how it occurs. Okay, so non-disjunction can also occur with the sex chromosomes. So remember that the sex chromosomes are not a true homologous pair in the sense that they don't code for the same information, they're clearly not the same size, and they're not the same shape. However, they behave exactly like a homologous pair in that they too get separated during meiosis, so that two of the gametes are going to contain the X chromosome, two of the gametes are going to contain the Y chromosome, that is for baby boys. But doesn't matter, even if this were another X, non-disjunction can occur in the sex chromosomes. And so here are some examples of non-disjunction that takes place inside the sex chromosomes or within the sex chromosomes. You can have the triple X syndrome. Of course, the infant or the individual will be female. They have an additional X chromosome. And again, like we said, these are very large kinds of changes, and so there tend to be some physical manifestations or symptoms that would go along with it. Some are more severe than others. So individuals who have the triple X syndrome can be slightly taller than average, and they tend to have more learning difficulties or learning disabilities. Jacobs syndrome is when the infant receives two Y chromosomes, and that individual will be male, because if there's a Y chromosome in the human genome, that determines maleness right there. And so by having two, the individual is still going to be male. They tend to be pretty normal individuals, but they tend to be slightly taller. Klinefelter syndrome, this one's more severe, so the child has two X's and a Y. They're male because, again, it's the presence of that Y chromosome that dictates that the individual is going to be male. And these individuals are sterile. They tend to have underdeveloped testes, and they might have more feminine features because they've got two of those X chromosomes. And again, remember, the problem with having too many or too few is now each of these chromosomes is doing what they're supposed to do, so you have more of the products of the X than what would be normal. And then having too few chromosomes. So here's an example where this O right here represents that there is no counterpart. So that would be one of these cells here that only received an X but did not receive anything else. And that would be Turner syndrome. The child is female because it's got that X chromosome. They too are sterile and have immature sex organs. So it can happen to the sex chromosomes. And here's again what's the telltale sign for these really is using a karyotype. So if there's any suspect that there might be something going wrong or with the pregnancy or something going wrong with the infant when the infant comes out, if you take a look at their chromosomes through a karyotype, you can see these kinds of abnormalities. Is there the correct number of chromosomes? And that's what karyotypes are so helpful for. The other thing that can happen on a chromosomal level that is also really detrimental to the offspring is when the chromosomes break. and that material that breaks off can be deleted. So it's completely lost along the way, and the chromosome then, reforms without any of that information. So that's really significant, and again, it can be determined by looking at a karyotype. And so here's an individual here where the top of chromosome 2 broke off and is lost. And so it's as though this individual here is lacking the second copy of all of those recipes. Now, if those recipes are really significant and they code for, features, physical features of an individual, then you can imagine that when that child is born, you're going to notice it right away in those outward physical features. So, genetic defects at this level are often very severe because you're missing large portions now of the DNA. So, that's looking at errors that can happen on a chromosomal level. So now we're going to get out of that. And so we're going to go away from errors. And for a while, what we're going to talk about is, okay, without it being that kind of situation, what's the normal handing down from generation to generation? So yes, meiosis can go wrong, but again, the probability of it is really, really low. And so things can go wrong, but what we want to focus on right now is what's happening when things go right. How do we pass down these traits from generation to generation? Okay, so what we're getting into now is called Mendelian genetics. And so when it comes to Mendelian genetics, we give Gregor Mendel a lot of credit because he was the first individual who stepped forward and started doing some research and looking at what is the probability. Is there something else going on within cells? Is it all just chance? Do we just look the way we look based on chance? Do you get a disease based on chance? Or, in fact, is there something going on? And Gregor Mendel was really the scientist, and he wasn't a scientist, he was a monk, but he was growing pea plants for the monastery, and in doing that, he noticed that when he was moving the pollen from the male plants over to the female parts of the plant, then he noticed that he got in very specific ratios the type of offspring. And so with that, he started working out that there was something going on inside of us that we were handing down from our children. And he put together that it comes in pairs. So this idea that our chromosomes come as a homologous pair is credit to Gregor Mendel. He put that together. And he also put together that you're going to hand down either this chromosome number 1 or that number 1, not both members of the pair. So when it comes to looking at this stuff, then we've got some language here to talk about. We often talk about a character or a trait out there. So you can have, when we're looking at trying to track genetics, we might talk about their eye color, the trait for eye color. And there are oftentimes different forms of that trait. And I think we've already discussed that these are called alleles. So of the different forms of a particular trait or gene out there, we call these different forms alleles. So you might have the blue allele for eye color, and you might have the brown allele for eye color. Okay, and so when we're looking at that, remember they lie on a homologous pair of chromosomes, And so if an individual has two different forms of the allele, like the brown allele and the blue allele, that individual is termed heterozygous. So hetero means different, and you're saying different forms, basically, of the allele. So an individual who has two different forms of the allele is called heterozygous. If you have two identical forms, so maybe over here this D represents your hair color and this is brown hair, and so here's brown hair. If you have two of the same forms of the allele, you're termed homozygous. So for each of these traits that we want to talk about, we talk about your genotype. And your genotype is your letter combination. And so we could say I'm a capital B, lowercase b, but that doesn't sound very sophisticated, and so instead I can say I am heterozygous for eye color. And I've got the brown allele and I've got the blue allele. Or I can say I'm homozygous for hair color, meaning I have two identical forms of the allele. Oops. Okay, so their phenotype is the visible traits, and genotype is the letter combination. So keep in mind that sometimes I can see some of these phenotypes. It doesn't always tell me what the genotype is. Having freckles is a dominant feature, and what that means is that I only need to have one capital letter, and if I have one capital letter, then I'm going to have freckles. So it's as though this other form here doesn't even matter. If this was a lowercase f, you would still see freckles on my face. However, so it's like this one doesn't matter. It does matter because, I'm going to pass it down to my children. But for something to be dominant, it means that you only need one form of it, and you're going to phenotypically see it. If something is recessive, what that means is that you have to have both recessive alleles in order for it to express itself. And that's what's true with chocolate labs. Chocolate labs, the only reason why they're brown is because they've got two recessive alleles. If one of these was a capital B and coded for black coat, then that dog right there would actually be black. The only reason why it's brown is because it has two recessive alleles, and that's what it means to be recessive. So when we deem a trait as being dominant and a trait as being recessive, that's what we're talking about. And this is what Gregor Mendel did, is he worked with pea plants. Pea plants produce a lot of offspring, so he could see if the numbers held up. They have lots of traits. They reproduce really quickly, so you can get to your seeds. And Gregor Mendel was basically running the greenhouses for a monastery, running growing the plants and growing the food for the monks that lived in the monastery. And he worked with pea plants. And what he found is that purple is dominant over white. And so P is a horrible letter to use in this case because it's really difficult to tell if this is a capital letter and that this is a lowercase letter. I would use something different. So when you're tracking genetics, try to use letters that are very obviously different in the capital form versus the lowercase form. This individual right here is the heterozygous individual. One capital, one lowercase. I've got two different forms of the allele. Here, the individual has two of the same forms, homozygous. Two of the same forms, homozygous. And because those are capital letters, we could say homozygous dominant, homozygous recessive, two lowercase letters. Heterozygous, period. You don't say capital or lowercase, just heterozygous.